This is Roger Green, host of the Surfing the MASH Tsunami podcast. Today, we're offering 5 conversations from episode 24, our discussion about looking back at EASL from 6 weeks after, plus a 5th discussion, which is a review of the question of the week from conversation 23. We've got Jeff McIntyre and PharmaNest CEO and founder Mathieu Petitjean for the interviews, and Mike Patel and Jose Willems for the discussion of the QOW. Just so you remember, the first question we asked Matt Petitjean was what his key takeaways were from EASL Congress 2024 pertaining to his business. Before answering, he describes the core services that his company offers, and I'm quoting here, PharmaNest specializes in digital pathology. 4 years ago, we put down the hypothesis that the histological phenotype of fibrosis should be quantified in a high-resolution, sophisticated way. He goes on to state their core proposition from MASLD, quote, the big idea here is fibrosis equals phenotype, end of quote. He then goes on to describe his offerings in greater detail before offering an underlying value of computed histology. Fibrosis is a continuous variable that is scored in discrete categories under the NASH-CRN model, which drives FDA analysis. Within this context, he answers the question by describing 3 kinds of MASLD clinical trial designs. The first earliest trials had a single pathologist reading histological slides. The method's not precise, the drugs weren't very good, so none were approved. The second set of trials relied on more rigorous methods for pathologists to read histological slides with multiple readers and robust adjudication system. And the drugs in the second set of trials were more efficacious so that NASH-CRN, while a blunt instrument, could adequately assess efficacy, which is why resmetirom is on the market today and others are coming. For the third set of trials, with even more robust drugs, but the opportunity to take a look at how they work differently, Matt believes non-invasive tests are likely to suffice and possibly even perform better for that specific purpose than digital pathology. As we've discussed over the last several episodes, EASL Congress 2024 was really a seminal, empowering event for the MASH and MASLD community. Listening to these 2 dialogues with Matt Petitjean and Jeff McIntyre gives you a sense of the many different ways that players could look at this and the impact that it will have on their businesses or their not-for-profits going forward. It's an important set of issues and I think really intriguing. So just sit back, listen, learn, absorb, and when you're done, join the conversation in our LinkedIn discussion group.
Mathieu Petitjean.
Just before I share my takeaways from EASL, I'm going to explain what we do so it's simpler as a way of setting the stage. So PharmaNest is a company that is specialized in digital pathology. And 4 years ago, we put down the hypothesis that the histological phenotype of fibrosis should be quantified in a high resolution, sophisticated way. And we also hypothesized that fibrosis looks different according to severity, which we all know, but according to other phenotypes, clinical phenotypes could be the gender, could be the etiologies that yields to fibrosis. So, the big idea here is that fibrosis equals phenotype. So we developed this tool, which requires highly sophisticated computational tools. We are a single fiber analysis company, and by single fiber, try to understand and anticipate the analogies with technologies such as single cell analysis. So we are both spatially resolved technologies, and all the data we generate on single fibers, we use them to find things that change as the phenotype change. So this hypothesis was original 4 or 5 years ago. It was not the common way of looking at scoring for fibrosis, and it happens it works very well, not only in liver, with other indications. So we do have now biomarkers for fibrosis. They are continuous biomarkers. They are under review by FDA and being used in phase 2 clinical studies. And what they do mostly is being able to identify changes within categorical stages. So, for instance, if you have a patient that is in an F3 plus stage that moves down to F3 minus, when you use categorical stages, that patient would not be a responder. It is going from F3 to F3 non-responder. And we all know that through biomarkers or other markers, we know the patient has improved. We resolve that challenge. We can measure changes inside categorical buckets. We are still based on biopsies. And with that said, I'll share a little bit what we took away from EASL. For me and then for my team, we debriefed internally. This was perhaps the most significant EASL that we've been through since even before COVID. COVID. And it's driven by several things. Obviously, the fantastic news that were reported at the late breakers. We had several studies with amazing antifibrotic and antisteatotic effects. So I think this was highly energizing for the field. For us, this was the big news, and we were excited to see that happen and to see, folks from fantastic biotech companies sitting side by side with us, and they were very, very happy to present those results. What we saw as well is the commercial deployment of Madrigal and resmetirom. I think they're doing a fantastic job. I was very impressed by the training sessions that hepatologists have been giving to really explain how to evaluate patients and how to put them or not to put them under resmetirom. I think this was extremely well done, and I don't think we've seen anything like that in prior EASL meetings. So these two things stand out. Now, all that said, we see as well, and this is particularly important for us, more and more convincing and significant results on non-invasive biomarkers, which we all know, biopsies as part of the criteria to enroll patients. And, if I'm a patient and somebody says, Mathieu, we're going to give you the choice to go into an investigational product or resmetirom, right? If you want to go into this investigational study, you have to take a biopsy. If you take resmetirom, you don't. That poses a true problem. Okay, is that patient going to consent to an investigational study? I don't have the answer, but for me, it's a big question mark that I think needs to be addressed. We can talk about that. And the other thing that we've seen is that the effects, the magnitude of the antifibrotic effects that were reported by some studies is so big that actually, who cares about finding patients that go from F3 plus down to F3 minus, right? The effect is huge. So I think categorical biomarkers such as the NASH-CRN that we thought would not be adequate to evaluate these studies work extremely well when the drug is strong and works well. So I think this is a lot of very good news for patients. And I don't know what will be in the coming months the position of regulatory agencies when it comes to keep or not to keep biopsies as part of the recruitment. My take is that we are currently in the generation 2 of clinical study designs. Generation 1 were the very first studies, the Intercept, the GenFit, the Falcons at BMS, the Gilead. Pathology was there. We had one pathologist at best instead of a panel. So these were generation 1. We are currently witnessing the results of the generation 2 studies where pathology has significantly improved, where there is a cohort of biomarkers to enrich the understanding of therapeutic interventions in NASH. And I think we're going to see pretty soon a third generation of studies where non-invasive tests will play a major role.
Now, when you said major role, do you mean to the exclusion of biopsy?
Possibly. Yes, we don't know, but I think we're going to we possibly are going to have to see them happen, and it's a very good news.
Roger Green.
I think for a patient. And clinical trials, On the way there, one of the things that you commented on that I think is important is that the NASH-CRN 4-point scoring system is, in fact, its own problem. I forget who, but somebody published last week a study using the PathAI-ML system that went back and looked at semaglutide data that had previously been defined as having no significant effect on fibrosis and said if you change it to a linear variable, the effect is about half a grade. And, in fact, it's demonstrably significant over the same time. So the ability to do the kinds of things that you do or HistoIndex or PathAI does will have some real value in generation 2. Now, tell me what that means for generation 3.
First of all, yes, in generation 2, having a continuous score that resolves what happens within categorical stages will be useful. Always. In particular, if we are looking for a moderate antifibrotic effect, if you have a GLP-1 and you are trying to see if that intervention on the short term impacts fibrosis, you need biomarker with a high detection threshold. So our automated methods can work. I think the same questions will apply when we will be studying combinations, right? If compound A and compound B are analyzed independently and then together, what's the difference in the fibrotic effect? You are going to need techniques with very high detection thresholds. But with compounds that have such a big effect as the one that were presented at EASL, I think categorical stage will demonstrate that the intervention works.
Yeah, FGF21s, for example.
Correct. Yeah, absolutely. The Falcon 1 and Falcon 2 studies, we analyzed them. Our data didn't demonstrate that there was a superior statistically significant effect. So same conclusion as the categorical stages. But what we presented 2 years ago at AASLD is that we have been able to identify phenotypes of fibrosis that changed. And those phenotypes, when we put them together in a score, we see that the score demonstrates a statistically significant effect. In other words, and dose-dependent effect. In other words, there is a favorable remodeling of fibrosis, but not sufficient during the duration of that study to move patients from one stage to another one. So either patients don't respond well or strongly or quickly, or the drug is not potent enough, but it works.
So let's go on to generation 3. Your point, I think, was that for drugs with profound effect, FGF21, will there be drugs? You say combinations and moderate effects might still need biopsy. Some of the most robust effects probably will not.
I'm not sure. I think these are open questions there. I think the first question is, what will regulators go for? What we see, and you know that very well, we see retrospective studies with non-invasive biomarkers with a significant amount of data that shows that a non-invasive test could be considered as primary endpoint for the studies. We see that happening. Our community will have to explain to FDA, as the community has done in the past, to convince FDA that NASH-CRN stages were the right endpoint for interim approval. They will have to do the same for non-invasive tests. The case would probably have to be very strong for F2, F3 moderate fibrosis patients. If we're talking to severe cirrhotic studies, I think the case will be easy to put. So perhaps a stratification between severe decompensated cirrhosis studies and those F2, F3 studies. I don't know. What I see for sure is that as exploratory endpoints, and definitely in the context of phase 2 studies, to really understand what's going on and enrich what we learned from non-invasive tests, I think the use of biopsies will always be positive. And then there is another way of looking at this. If we believe that these treatments need a personalized approach, if we say this disease needs to be addressed with a personalized medicine perspective, the fact that we have a biopsy is very valuable. In that case, we need to make sure that we get the most out of this tissue. And modalities such as oncology do that very, very well. So if we believe that a personalized approach is needed, then biopsies will play a big role.
Turning the question about 30 degrees, the question might not be whether personalized approaches are needed. It might be part that and part, are we capable of creating personalized approaches that bring value, right? In a world where you've only got 3 agents to work with, that's unlikely to happen. But as you get more agents, then in fact, you not only can do a better job of figuring out what phenotype or category a patient falls into, but then also what do you want to do about it? Or how would you handle it differently?
And these hypotheses are not incorporated in any study that I know right now. But I think it's in the debate, should we or should we not keep biopsies in the context of MASH studies? The case could be made that biopsies will be useful not only to stage fibrosis and the related histological endpoints, but also to personalize or investigate possible personalized medicine approach or to stratify patients in different ways.
Okay, so let's play with that for a minute. If I think about the history of diabetes, there was a point at which everybody got metformin first line. And then after that, patients who did not do well on metformin or whatever point metformin did not work adequately went in different directions. If I think about that in the context of MASLD and MASH, maybe where I get to is a bunch of first-line therapies that I can give without personalization. But then if the first-line therapy fails, then at that point in time, I might want to conduct biopsies simply to understand enough about the patient on a personalized basis that I can prescribe exactly the right regimen for them. Is that kind of where I hear you going, or is that just my thought?
Well, I'm not sure I'm the best person because I'm not a physician, so it's always difficult to ask me how to manage your patients. Thinking logically, the first thing that I would say, okay, if you prescribe first-line therapy and you said if it's not working, right, how do you assess that? So it's going to be assessed based on non-invasive tests. Is that going to be assessed on ultrasound MRE? So I think that would be the first question. How do you assess and how long do you need to wait to assess that before you prescribe to something else. Because we're not talking about weight here. It's a little bit more difficult to measure. But I think if they are non-responders, or I should say slow responders rather than no responders, where there is urgency to address the metabolic etiologies, I think what you propose is very reasonable. But I don't see the avenues that could be proposed today.
We don't have options. I mean, back at oncology, how long did we have to be in oncology and how many different kinds of drugs did we have to have?
That's correct.
Or interventions in general before it made sense to personalize to that degree?
I think there are two considerations in terms of time. As you said, is how long it took us to substratify breast cancers. It didn't happen overnight. And the other thing which a lot of people ask me says, how long will your biomarker, we're a digital pathology biomarker, how long will it take for this biomarker to become a surrogate biomarker so it can be used instead of the NASH-CRN? Well, let's go back and take a look to A1C. It took a lot of time to turn A1C from a biomarker to a surrogate biomarker, and for all the studies to use that as a surrogate biomarker. So it's going to take a lot of time. We are currently running 2 outcome studies, retrospective outcome studies, to really be able to establish the link between our biomarker and clinical outcomes. One is an international study. The other one is a domestic study. We will actually retrospectively analyze the NASH-CRN database too. Doing retrospective studies will accelerate the data that we generate. But having said that, being a surrogate biomarker is a extremely demanding value proposition that needs a lot of data. So we'll see if we can go as fast as this new generation 3 trials.
And now, back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please put them in the review section of the page from which you downloaded this conversation, or send an email to questions@surfingmash.com. Also, remember to answer our questions of the week so that you can have a chance to hear panelists discuss them and be part of that discussion and recognized on the program. In our next episode, Maru Ranella joins Jorn, Louise, and me to discuss the recent expert recommendations on resmetirom paper published in CGH. Until then, stay safe, surf on. We'll see you on the podcast. Bye-bye now.